I'm Iain Mathieson, and I'm going to talk a little bit about our work using ancient DNA to learn about human evolution and natural selection. So ancient DNA, as many people will be aware, has really made a huge difference in our ability to use genetic data to make inference about the history of past populations. Some people describe ancient DNA as cheating because rather than use complex statistical methods to make inference about the past, we can really just look at it directly. Here I'm showing you one of the earliest examples, where by sequencing only 2 ancient genomes from around 8,000 years ago, the authors were able to infer that subsequent and present-day European populations are an admixture of at least 3 different ancient groups. Since then, many papers have refined models of European genetic history, as well as cast some light on other parts of the world. In principle, ancient DNA should be equally transformative for studies of natural selection and the evolution of functional parts of the genome. By measuring changes in allele frequency over time, we can directly observe evolution in action, almost as though we were performing an experiment. However, ancient DNA-based studies of selection have been slower to take off relative to studies of demography. A large part of that is that they require much larger sample sizes. To see why that's the case, here's a cartoon of your genome. So you have 2 haplotypes, one of which you inherited from your mother and one from your father. And they inherited them from their parents and so on. If you think about the tree of all your genealogical ancestors, then any single position in your genome traces through one of the exponentially many possible paths in that tree, shown here in red. If you look at another position in your genome, then because of recombination, the ancestry of that position will take a different path through the tree, and therefore give you information about a different set of ancestors. Thus, by looking across many positions in the genome, even the genome of a single individual, we learn about a sampling of all of that individual's ancestors. So we're not just learning about n equals 1, we're actually learning about a large proportion of the ancestors of that individual. And so that's why we're able to make what seem like quite sweeping inferences about history just based on the genomes of 1 or 2 individuals. However, if instead we're interested in studying selection or the evolution of a single locus, for example here in blue, then we aren't able to average over the whole genome. So when we have n equals 1, we really do have just one observation. And that means that in order to study the evolution of individual loci, we really do need much larger sample sizes. Fortunately, we do now have quite large sample sizes. Here's the cumulative number of individuals with ancient DNA published up through the end of 2019. As you can see, it was really only around 2015 that we started to have sample sizes in the hundreds that we needed to really start to study selection. Now, the majority of those samples are from Europe and Western Eurasia, and I'm really only going to be talking about those regions. That's not necessarily because I think they're the most interesting places in the world, but it's just really because it's the only place where there's currently enough data to do the kind of studies that we are trying to do, although that is beginning to change. So, given all these samples, what do the data tell us? Here I'm showing you an MDS plot of somewhere around 1,200 ancient Europeans. So you can think of this as a figure that's summarizing genetic variation among these individuals into 2 dimensions. The coloured points represent ancient individuals coloured according to the time period in which they live, here in the timeline at the bottom of the slide. This covers the entire 45,000 year period that modern humans have been present in Europe. Grayscale points represent present-day European individuals taken from the 1,000 Genomes Project. And the first thing you notice is that the majority of ancient Europeans fall outside the range of present-day Europeans. That tells us that most of the people who historically lived in Europe did not genetically resemble present-day Europeans. If we look at the earliest time periods, starting with the oldest individuals here in the early Upper Paleolithic in gold in the centre of the plot, and they appear in the centre of the plot because they're really symmetrically related to all the subsequent populations. These individuals really are quite closely related to the out of Africa population that is ancestral to all present-day non-African populations. As we move through the last glacial maximum, or LGM, we see a drift here, and once we reach the Mesolithic, say around 10,000 years ago, genetic diversity is largely laid out along this cline. In fact, this cline corresponds to an east to west geographic structure established during the post-LGM repopulation of the continent. Starting around 8,000 years ago, we see a dramatic shift in ancestry coinciding with the agricultural transition. This shift is associated with the movement of people from Anatolia into Europe, bringing with them both the new technology of agriculture and this new type of ancestry. Hunter-gatherers are still present and they do admix with the farmers, contributing something like 5 to 20% of the ancestry of the Neolithic population. However, that population, as you can see, still does not look genetically similar to any present-day European populations. In fact, it's not until the Bronze Age, starting around 4,500 years ago, that we see additional movement of people into Europe, this time from the Eastern European steppe, which contributes perhaps around 50% of the ancestry of subsequent populations. At this time, we start to see ancient individuals who look genetically like present-day Europeans, although the geographic structure of these populations doesn't necessarily correspond to the structure we see today. So on a broad scale, that's what we see when we summarise changes in ancestry looking over the whole genome. And this historical interpretation has been established by many groups over the past 5 to 10 years. The next question is whether there are particular variants, genes, or parts of the genome that don't fit these patterns. If we can identify loci that aren't behaving like the genome-wide average, those are good candidates for genes that are evolving non-neutrally. So that's exactly what we did in this paper published in 2015. Each point on this figure is a single SNP ordered according to genomic position. On the y-axis is the log p-value, testing the null hypothesis that the frequencies of that SNP in ancient and modern populations are consistent with the genome-wide average. So when you see a big spike, it means there's good evidence that that part of the genome is evolving non-neutrally. Now in this analysis, we're really only looking at samples from the past 8,000 years, the period of time during and subsequent to the introduction of agriculture. And this is exactly the sort of figure you'll have seen if you've seen the results of genome-wide association studies or other gene mapping experiments. Fortunately for us, unlike most genome-wide association studies, most of these hits occur in or close to genes that we have a pretty good idea of the function. Here are the genes that we identified as being likely to be affected by each of these signals. And if we ask what these genes do, it turns out that they fall into three main categories. So there are genes associated with diet, genes associated with skin pigmentation, and genes associated with the immune system. So that makes intuitive sense if we recall that we're looking at selection after the introduction of agriculture. People are adapting to a new diet, moving into a new environment with less UV radiation, perhaps being exposed to new immune challenges due to new environments, denser populations, or the presence of domesticated animals. One of the powerful things about having these kind of data is that we can look at these individual signals and really start to drill down and identify the timing and location of selection. If we look at the lactase persistence locus, this is a well-known variant that confers the ability to digest lactose as an adult. It's actually the strongest known signal of natural selection anywhere in the entire human genome in any population. So in that sense, in this experiment, it's really a positive control. Here, we've used ancient DNA from different parts of Europe to jointly estimate the allele frequency trajectories and selection coefficients. We find that in Britain, Central Europe and Iberia, the allele frequency starts to increase perhaps 3,000 to 4,000 years ago, consistent with a selection coefficient of 2 to 2.5%. That's actually several thousand years after cattle domestication and the introduction of dairying. Conversely, in Italy, the frequency always remains low, although non-zero, and we estimate that the persistence allele was never actually under selection at all. This raises an interesting question about why the selective pressure would be different in different parts of Europe. I think that's something we will investigate further in the next few years. Staying with selection on diet-related phenotypes, we see another strong signal at the FADS locus, which seems to contain extremely interesting genes. So what do these genes do? The immediate function is to catalyze the synthesis of long-chain polyunsaturated fatty acids from short-chain precursors, illustrated here by Fumagalli et al. Now, these long-chain fatty acids are critical for many functions, for example, the construction of cell membranes. As Fumagalli et al. Pointed out, if you eat a diet that's relatively plant-based, containing a lot of short-chain fatty acids, you may need more enzymatic activity to synthesize the long-chain versions. Conversely, if you eat a diet that's relatively high in meat and marine fats, you can obtain a lot of the long-chain acids from your diet, and you don't need to synthesize so much. So, the hypothesis is that this FADS allele is kind of a genetic tuning switch, allowing us to adapt to different types of diet. These variants have a lot of downstream effects. If we look at the UK Biobank, we find that they're very significantly associated with blood cell phenotype. They're also one of the most significant genome-wide associations for lipid levels and some types of hormones. Just as with lactase, we can look at the timing of selection. So here on the x-axis is date in years before present, and on the y-axis is the frequency of the derived allele. That's the allele that we would hypothesize would be associated with a plant-based diet. Here we split up individuals by ancestry. You can see that hunter-gatherers in green don't carry the derived allele, early farmers in blue do, at a frequency of around 35%, and it really starts to increase in frequency starting around 4,000 years ago, actually thousands of years after the introduction of agriculture. I'm not showing this here, but we do find, unlike with lactase, that this increase is consistent across all parts of Europe, estimating the selection coefficient to be around 0.8%. That's very interesting, and it's clear from this direct evidence that the allele has been under selection in the past 10,000 years in Europe. But it actually turns out that the history of selection at this allele is much older and much broader than that. The derived allele is very old. In fact, part of the haplotype is shared with Neanderthals, and it actually seems to have been selected very anciently in Africa, before the Out of Africa event. It's largely fixed in present-day African populations, and interestingly, early Out of Africa populations seem to have reverted to the ancestral allele, potentially because the large megafaunal populations in Eurasia would have enabled a more meat-heavy diet. The ancestral allele was fixed in Europe by the Mesolithic, and then, as we've seen, the derived allele was selected in the last 10,000 years after the introduction of agriculture. Here, the CEU population represents present-day Europeans. It also seems to have been selected in parallel in East Asia, also following the independent development of agriculture. So this variant really has been around a long time, under selection and helping people to adapt to new environments. It turns out that this selection may still be ongoing today. So the top panel here shows the result of the selection scan zoomed into just the region around the FADS locus. Again, each point is a SNP, and the y-axis shows you just the p-values for the selection test. You can see the FADS genes here in the lower panel. The center panel shows the result of a genome-wide association study for fertility. Here, the p-value is whether each SNP is associated with number of children in a meta-analysis of around 750,000 European adults. We find that the derived allele is associated with an increased number of children that would correspond to a selection coefficient of about 0.7%, remarkably close to what we estimated in the historical selection scan. Since this allele is associated with increased fertility and therefore potentially fitness, it seems plausible that the historical selection we observed might still be ongoing. Here's another piece of evidence. On the left is the increase in frequency of the allele over the past 10,000 years as measured from ancient DNA, while on the right is the change in frequency as a function of birth year in the UK Biobank, small but consistent with the historical rate of change. One final interesting observation is that there's no overlap between any other fertility GWAS signals and any other scans for historical selection. So for example, lactase persistence, which was so strongly selected in the past, is not still under selection today. Conversely, none of the other variants that affect fertility today were under selection in the past, at least in this largely Northern European population. Now, so far, all of these variants that I've talked about have been single variants with relatively large effects. Really, we're looking at strong sweeps, whether on new mutations or standing variation. But it's widely believed that a lot of adaptation is polygenic, and we can also find examples of that with ancient DNA. One of the classical examples of polygenic adaptation is skin pigmentation. Light skin pigmentation in Europe is largely believed to be an adaptation to low levels of ultraviolet radiation, although the exact selective mechanism remains unclear. Even in Northern European populations, it's a highly polygenic trait, with something like 180 genome-wide significant loci in the UK Biobank. So what's plotted here is, for each point representing an ancient individual, the proportion of those loci where that individual carries the pigmentation increasing allele. And what you can see here is that as we go further back in time, the proportion tends to increase. This increase is highly statistically significant when compared with the rest of the genome, and provides evidence that these variants as a whole have been under selection. In fact, it turns out that this change is driven by a relatively small number, perhaps 5 to 10, variants with relatively large effects. Now, one caveat about this analysis is that we can't make inference about the phenotype of these ancient individuals. This analysis is telling us about the trajectories of the variants that are polymorphic and associated with the phenotype today, and providing evidence that those variants were under selection, but it can't tell us about any other variants that we might not be aware of, so we can't actually make phenotypic predictions. In any case, we couldn't test such predictions because, of course, skin, being a soft tissue, is not preserved for these ancient individuals. There are a few phenotypes we can measure in ancient individuals, mostly skeletal, and in a paper last year we did test genetic predictions of some of those phenotypes. On the left here is a genetic prediction for a stature constructed from SNPs associated with height in the UK Biobank. Again, each point is an individual, and we find significant differences in predicted stature between the early and late Upper Paleolithic and between the Neolithic and Bronze Age. If we look at measured stature on the right, not in the same individuals but at least in individuals from the same populations, we see qualitatively similar patterns. So at least in this case, the genetic predictions are consistent with the phenotypic changes. Now, this analysis is not quantitative, and I think there are a number of caveats remaining in the interpretation, particularly issues to do with correction for population stratification. I do think it's promising, and we hope to pursue these approaches at least to understand the evolution of skeletal phenotypes. So that's the work I wanted to present today. I think in the near future we'll see more data from parts of the world outside Europe, hopefully led by researchers from those parts of the world, and we're already starting to see that in places like East Asia. That will allow us to test hypotheses about the way in which humans respond to changes in environment. Particularly in the case of examples like the introduction of agriculture, one of the few repeated experiments in human evolution. And finally, I hope we'll see these sort of approaches more widely applied to non-human species, both wild and domesticated. Ancient DNA is really turning out to be a very powerful tool for learning about evolution. I'd like to thank my group at Penn, as well as external collaborators and co-authors on the work I've presented, and of course all the funding sources who've supported this work. I also want to thank SMBE, both for this award and for the journals and the meeting which have such an important place in the field. I very much hope we'll be able to continue those meetings in the future, and I thank you very much for listening.